So when we left off on Tuesday, we were talking about this rather non-informative figure that looks at the role that Phytochrome plays in regulation of gene expression. And let me just remind you that in this particular figure, the only role that Phytochrome is playing is that it happens to be degraded by COP1. But even that is an inconsistent picture because the text describes to us, and it tells us in this picture, that in the light, COP1 is exiting the nucleus. That allows these transcription factors to drive light-dependent gene expression. Those same transcription factors are ubiquitinated by COP1 at night. Because remember, COP1 is an E3 ubiquitin ligase, the thing that sticks ubiquitin specifically onto target proteins. Okay, so the role that Phytochrome is playing in this process, it should be clear that it's not clear. That we really don't know what's going on in any great detail here. Okay, so the receptor end of Phytochrome-dependent signal transduction pathways are very well understood. We have crystal structures of Phytochrome. We understand the photocycle between the red and the far red reversing forms in exquisite detail. I mean, they've measured structural changes in the femtosecond time range. So we really know what's going on there. But even the next steps, what Phytochrome directly interacts with to trigger various types of Phytochrome-dependent responses, we really don't know. Right? So there's a lot of unknowns in what we're dealing with Phytochrome. And as I said before, we're going to encounter that regularly through the rest of the semester. Okay, any questions about this before there's one more thing I want to say about Phytochrome to sort of bring this all back together, but any questions about this before we go on? Okay, so one of the things that we mentioned back at the very beginning of the lecture was that the red, far red reversibility that is the hallmark of Phytochrome because of the way the pigment protein complex works. The question was, under how many circumstances in the real world is red, far red reversibility physiologically relevant? You know, so for example, in slowing down stem elongation, does the plant ever experience far red light after red light that should be a signal to the plant to stop stem elongation? And the answer is probably not. So the question is there a relevance, physiological relevance, to the red, far red reversibility? And there is one circumstance where that is the case, and that is the response of plants, at least of sun plants, to shading. The general response of sun plants to shading is that they grow taller. Not particularly surprising. If plants are competing for light, then if plants are shading each other, then the way the plant can win the competition for available light is to grow taller faster. So in sun plants, plants that are grown in shady environments tend to be taller, they have longer internodes, they have fewer leaves than plants that are grown in the shade. If we look at shade plants, the difference is relatively small. And that's not surprising because they are evolutionarily adapted to living in shade environments. Where sun plants, when they're in the sun, they're growing optimally. When they're in the shade, they want to get into the sun. So the question is what controls this, and it shouldn't be particularly surprising that it's the red, far red ratio of the light. Because remember that under the canopy, the red light's been absorbed, and there's far red light is what's dominating under a canopy. So the red, far red ratio of the light, and therefore the ratio of PFR to the total Phytochrome is going to change. It's going to decrease as one goes to sunnier environments. So this is the one example of where the red, far red reversibility of Phytochrome plays a really important physiological, ecological role. So let's step back to finish this off. is this what Phytochrome evolved to do? In other words, is the red far red reversibility something that's been selected for because of sun shade adaptation? What do you know that might be useful to that, to answering that question? Does Phytochrome exist in other photosynthetic organisms besides plants? Is there Phytochrome in mosses? Yeah, there is. Read it. It was in the textbook. Is there Phytochrome in algae? There's Phytochrome in cyanobacteria. So the most primitive types of photosynthetic organisms have Phytochrome in it. So I think what that tells us is Phytochrome did not evolve to respond to sun and shade environments. The characteristics of Phytochrome, that red far red reversibility, was an inherent property of the molecule at the time it evolved. And it evolved as a light sensor, not a red far red reversible thing. That was part of what the molecule did. But it wasn't necessary for what it was selected for back probably several billion years ago. But it happens to work pretty well as a shade sensor under these circumstances. So this is one of those evolutionary questions where we do have some pretty good information that says, no, it's unlikely that Phytochrome evolved because it's red far red reversible. But some physiological responses can take advantage of that. Okay? All right. So, whoops, that's not what I meant to do. Sorry. Let's switch gears now and talk about blue light receptors. And a good place to start is with the question that about half of you asked in your reading questions that I looked at this morning. And that is there, if we have red light receptors, the phytochromes, and if we have blue light receptors, the cryptochromes and the phototropins, is there a specific difference in the way these different photoreceptors are used based on the wavelengths of light that they absorb? Anybody want to make a comment on that? So we've got this class of compounds that controls light-dependent responses that absorb red light, and we've got another class that absorbs blue light. Were some of those selected because they were the blue light receptors selected for because they absorb blue light, and the red light receptors selected for because they absorb red light? How are you going to answer that question? What do you need to think about in order to answer that question? What both of them do. Okay, so we haven't talked about the blue light receptors yet, but you've read about it. Are they completely distinct, or are they similar sorts of things? A lot of them are similar. More important question. Are there any of the responses that you can think about that phytochromes do that depend on red light that couldn't be induced by blue light, or vice versa? In other words, is there something important for any of the responses about red light versus blue light? We just saw one, right? The red far red reversibility is not something that would work that well in blue light because there aren't similar changes in the spectrum in the blue region underneath the canopy. But chloroplast movement. We're going to talk about chloroplast movement today with blue light receptors. Is there any reason that chloroplast movement should be driven by blue light and not by red light? It does have a higher frequency. So? Actually, blue, the shorter the wavelength, the more highly it's scattered it goes through. So it doesn't actually pass through the leaf better. Red light passes through better than blue light. Okay, I'm not going to answer the question today. That's the study. That's also today's study question. So I suggest you take a look at it and think about it, because something like this would make really good fodder for a final exam question. Okay, so to think about the blue light receptors then, the context of them is the same as the red light receptors. They are triggering light-dependent phenomenon, in this case in response to blue light rather than red light. One of the classical phenomena is one that was originally described by Charles Darwin and his son Francis. Dark-grown oat coleoptiles will bend towards the light. One of the interesting things that they observed is that the part of the plant that is responding to the light is in the tip of the coleoptile. The part that's bending is down here. So this was the first indication that there must be some sort of a mobile signal in the plant that carries information from where it's received to where the response occurs. Right? And we'll see in the lecture on next Tuesday when we start talking about auxins. Auxin is that mobile signal. Okay, so one of the things that physiologists like to do is these action spectra. Remember that an action spectrum is basically measuring some physiological response as a function of wavelength. Why do we do action spectra? Why does a physiologist measure an action spectrum? What good is it? What kind of patterns? It's a pretty picture, right? What does that tell us? Stephen, what does that tell us?
Well, like to bring about like a response, you have to have a certain wavelength.
Yeah, okay. That's the information that's on the graph basically, right? So we've got wavelengths. Certain wavelengths of light are more useful at producing responses than other wavelengths of light, right? But what does that tell us? What does this picture tell us? Is this Phytochrome? How do you know it's not Phytochrome? Oh, okay. So what does this tell us about the blue light receptor? It's a different receptor, and where does that receptor absorb? In the blue, right? This is the absorption spectrum of the acceptor. In the same way that the spectra that we saw for Phytochrome identify the absorption properties of the pigment that's in Phytochrome. This identifies the absorption properties of the pigment that's in the blue light receptor that's controlling coleoptile bending. Okay? So in a physiological sense, the action spectrum gives the person that's studying it the characteristic absorption spectrum of the molecule, of the pigment, that is actually doing the absorption. So when physiologists look at this spectrum, they immediately get confused. And the reason they get confused is because it is not unique to a single type of molecule. When this was first observed back in the 1940s, people immediately said carotenoids, because this shape, this typical 1, 2, 3 fingered spectrum, is characteristic of most carotenoids. Beta-carotene has a spectrum that looks very much like that. So the original thought was that carotenoids were the light receptors for blue light responses. But there are other molecules that have similar absorption spectrums. For example, flavins. Flavins, the place that you've encountered them is in the citric acid cycle. One of the enzymes, instead of being NAD, NADH, uses FAD, flavin adenine dinucleotide. So it functions similar to NADH. So they're flavins, and it turns out that coleoptiles tend to be rich in flavins. Riboflavin, you've heard of. Riboflavin is a vitamin. right? So there are other molecules that have this absorption spectrum. So when this action spectrum was first identified, it wasn't really clear from it what the molecule that's involved in the absorption is actually is. Okay. So when we talked about photomorphogenesis, one of the things that we talked about last week was the idea of phototrophic responses. And that means response to directional light. So for example, the decrease in stem elongation, the light-dependent decrease in stem elongation that happens in dark-grown seedlings, that is not a directional response. The light can come from anywhere. But when we talk about phototropisms, the responses are to light. So if the coleoptile is bending this way, it's because the light is coming from this side. If you gave the light from the other side, it would bend in the opposite direction. It's a directional response. And one of the things that you should be thinking about is what's the purpose of this directional response? Why would a plant even want to do this? Now, you don't need to answer it now, but you certainly should be thinking about that as you study this section. So that was the coleoptile is a monocot. Here's a Arabidopsis seedlings. It's a dicot. So obviously bending in response to light that's coming from the right. This is the high 4 mutant that the textbook talks about. We'll come back to it in just a little bit. A mutant that under directional light does not respond by bending the coleoptile. Okay, so this is obviously some mutation that is affecting somewhere in the signal transduction pathway between the receptor and the response. And we'll figure out what that is in just a minute. Okay, the response to stem elongation and this is only really in dark-grown seedlings. We talked about this as a Phytochrome response on Tuesday. Red light will relatively quickly slow down the stem elongation of dark-grown seedlings. It'll trigger that conversion from etiolated seedlings, that is no chlorophyll, hypocotyl hook, no leaves, rapid rates of stem elongation. Red light will trigger that transition. Blue light also does it, but the difference is red light, the Phytochrome-dependent response, is tens of minutes. The blue light response, in terms of slowing down stem elongation, is less than 1 minute. So 2 important things that come out of this observation. One is that both red and blue light control this. And we're going to see that there are many phenomena where there's overlap between red light and blue light. And you should be thinking about why that might be useful for the plant as well. But the other thing that we discussed last time is the response time tells us something about the signal transduction pathway. So it tells us that for whatever reason, the blue light signal transduction pathway that slows stem elongation is faster than what's happening with Phytochrome. So if we look at the rates of stem elongation following turning the blue light on, we can see that after a lag of about 30 seconds or so, there's a pretty rapid decrease in the rate of stem elongation. And this would be 10 times slower if we were talking about a red light response. There'd be a much longer lag for the Phytochrome. Now, another thing that people have observed in the cells that respond, whose growth rate responds to blue light, is a blue light dependent change in membrane potential. So if we see the membrane potential going from minus 150 millivolts up to minus 60 millivolts. What does that tell us is going on? What is the endpoint of the signal transduction pathway that's causing these changes? How do we change the membrane potential of a cell? Say that again. Be more specific. Let's be more specific yet. Well, it could be K+. Could it be sucrose? No, it could not be sucrose. Why not? Not charged, right? It's got to be something that's charged. So you're correct. It changes. There's got to be changes in the solute permeability of the membrane, but that change has to be for charged species, right? So these changes here represent ions moving into or out of the cell. So it tells us that somewhere between one of the responses of blue light to blue light is changing the permeability of the plasma membrane to some ions, and that may be in some way related to what's happening to the decrease in stem elongation. The change in the ion concentration, for example, if it was calcium. Calcium is a common component of many signal transduction pathways. So it could be calcium flux that then triggers something to do with the decrease in the rate of stem elongation. Okay. One of the areas of physiology that responds to blue light that's been studied the most is regulation of stomates. And obviously there's a lot of information in the chapter about stomatal regulation. So let's think a little bit about this. We remember from our discussion of stomates when we were talking about photosynthesis and gas exchange that it's changes in the turgor of the guard cells that causes the opening and closing of the stomatal pore. So what were the things that we discussed back when we were talking about stomates as being environmental signals that regulate stomatal opening? What sorts of things are going to have effect on the opening and closing of stomates? Blue light. That's a good one. That's the chapter we're reading. What else? Potassium will be what changes, but the cell is not responding to potassium differences. What other things can cause the stomates to open and close? No, not wind. What would wind affect that would cause the stomates to close? Yeah, water potential. Remember the chapter that we talked about stomates in had to do with regulation of plant water content. That when the plant could not replace the water it was losing by evaporation by water from the soil, the plant's response was close the stomates. So there's one other thing that people have studied that we haven't talked about, and that's carbon dioxide. Changing the carbon dioxide concentration also causes the stomates to open and close. High CO2, stomates close. Low CO2, the stomates open. So one of the things we want to try to sort out is how do these different regulatory factors in the environment actually control stomatal opening. And it should be clear to you that these two might be very hard to distinguish. Why? Why would blue light and CO2 concentration be hard to distinguish from each other? Say that again. You can't see CO2, but you can measure CO2 relatively easily. Yeah, that's basically it. So if you shine blue light on the plant, is the plant going to do photosynthesis? If it does photosynthesis, won't the plant lower the internal CO2 concentration? Yeah. So there's an indirect effect of blue light on CO2. So when you shine blue light on the plant and you see the stomates opening, are they opening because of the blue light? or are they opening because the CO2 concentration is decreasing because blue light is inducing photosynthesis? Right. Okay, so it's not a trivial thing to sort out, and there's been some pretty clever experiments that have been done to do this. Probably the most common one is to illuminate the leaf with saturating red light. Okay? Saturating red light in terms of photosynthesis. Okay? So that means photosynthesis is going as fast as it can. What happens if we turn on blue light to the rate of photosynthesis in a plant that's already saturated with red light? It'll stays the same. That's right, because it's already saturated. You add more light, can it do any more photosynthesis if it's light saturated? No. Okay? So the experiment is to illuminate the leaves with saturating red light and then turn on, actually this is relatively weak, blue light, and measure the changes in the stomatal aperture. And what you see is that the blue light causes an increased opening of the stomates even when photosynthesis is saturated. So now we have separated out these two effects. There's no changes in CO2 associated with the blue light because photosynthesis doesn't change if it's red light saturated. So the blue light effect must represent an independent effect of the blue light on stomatal aperture. What about red light? Could red light be affecting the stomates by some other mechanism than CO2? Have we heard about any red light receptors that might affect things that cells do? Come on, you guys. What did we talk about on Tuesday? Phytochrome. Is phytochrome a red light receptor? Could phytochrome be playing some role here? It could. Yeah, absolutely it could. Guard cells are really cool for sorting out these things because the phytochrome genes are not expressed in guard cells. Guard cells don't have any phytochrome. They have the genes, but the genes are never expressed. So that makes guard cells a really nice system to study blue light effects without the complicating effects of phytochrome. So lots and lots of work has been done on blue light effects in stomates to try and sort out what's going on. As we'll see, what's happening in stomates is probably different than what's happening in stem elongation or chloroplast movements or things like that. Okay, so here's the action spectrum for the blue light. It's got a pretty similar shape to what we saw for the coleoptile bending, so it falls into this category of blue light receptors. So some simple experiments to try and sort out what's going on in the blue light dependent swelling of the guard cells. One clever thing to do, although it's very, very challenging to do, is to make guard cell protoplasts. So basically, peel the epidermis off the surface of a leaf. Remember, the guard cells are embedded in the epidermis. And then digest the epidermal cells with some mix of cellulases and stuff like that. The thing that's good about guard cells is they have a different type of cell wall than the rest of the epidermal cells that are a little bit more resistant to the cellulases. So you can actually isolate guard cell pairs that are still around the stomatal pore relatively easily and then digest those a little bit further and get real guard cell protoplasts. Okay, so the simple experiments: put the guard cells in blue light, compare them with the dark, and see what happens. No big surprise. The volume of the guard cells swell in blue light relative to the dark or relative to red light on. So again, saturate the effects of photosynthesis. Oh yeah, one important thing. Guard cells are different than every other epidermal cell. Epidermal cells do not have chloroplasts. Guard cells do. We're going to see the role that these chloroplasts play in a lot of what we think is going on in terms of guard cell responses to environmental things. Okay, so I'll comment on the role of guard cells the role of chloroplasts in guard cells just a little bit, because it's important to have this sort of physiological perspective. Unlike the mesophyll cells, where in normal light conditions, the mesophyll cells are net exporters of carbon. They take the products of photosynthesis and convert it into sucrose or some other transported molecule and take it out of the cell. Guard cells are not net exporters of carbon. The amount of photosynthetic product that is made in the guard cells is not sufficient to supply the guard cells' carbon demands over 24 hours. Okay, so guard cells remain net importers of carbon from the sources in the plant. So you might ask the question, why do guard cells have chloroplasts at all? If they can't make enough to help them to meet their needs in terms of carbon, why do they have chloroplasts at all? Well, it should be pretty apparent that photosynthesis is playing an important role in what guard cells do. Right? But keep in mind that a guard cell is not the same as a mesophyll cell in terms of its response to light. It has fewer chloroplasts. The chloroplasts aren't as efficient at doing photosynthesis. They can do photosynthesis. They can make sucrose and starch, as you've seen. But they're not the same as mesophyll cells. Okay, so when we put the guard cell protoplasts into blue light, the protoplasts expand. A very interesting observation was that vanadate is a very effective inhibitor of the plasma membrane proton pump. So the primary active transport pump that's present in the plasma membrane of all plant cells is inhibited by vanadate. And so when you add vanadate to the guard cell protoplasts, the swelling of the guard cells is inhibited. So right away, that is an important piece of information. It's telling us that the activity of the proton pump is important in guard cell swelling. No big surprise there. I mean, the proton pump is what powers all the ion transport across the membrane. So if we inhibit the proton pump, presumably whatever is contributing to the swelling can't happen. Another experiment that's been done is simply to look at the pH of the medium that the guard cell protoplasts are suspended in. When you turn on blue light, you see the medium is acidified. It becomes more acidic in the blue light, and the amount of acidification is light intensity dependent. Right? So the more blue light, the more acidification. That presumably means the brighter the light, the more the stomates will open. So there's a light dependence to this that we would expect to be there. Okay, so it's pretty clear that one end of the signal transduction pathway that begins with the blue light receptors in guard cells is changing the activity of the plasma membrane proton pump. And then that proton pump is presumably having some effect on the distribution of other solutes in the cell. And initially, as Michael mentioned earlier, potassium plays an important role in this. It's been known for a long time that acidification in guard cell protoplasts is accompanied by potassium uptake by the guard cell protoplasts. So there's an initial transport of potassium into the guard cells, and associated with the potassium uptake, if potassium uptake is going on, that's going to bring more positively charged things inside the cell. That should change the membrane potential. And yet the membrane potential goes through a transient, but it goes back to normal pretty quickly. But as we see, the proton pumping that's going on in response to a pulse of blue light lasts for a long time, 10, 20 minutes, something like that or more. Right? So the membrane potential may change transiently, but the the proton pumping goes on for some time. So that means the potassium uptake must be counterbalanced by some other ion. And so there's two other ions that contribute to the counterbalance. There's uptake of chloride. So at the same time the cell is taking up potassium, it's also taking up chloride. But the other thing that happens is it accumulates malate. Malate is the anion of malic acid. We talked about malate when we talked about C4 photosynthesis. So it's a compound that can be produced from starch in the chloroplast. It can be produced from triose phosphate in the chloroplast. So malate and chloride are the main counterions that keep electrical neutrality inside the cell as far as balancing the potassium uptake. Okay, so the interesting thing about this is that potassium uptake, if we look at the things that contribute to the osmotic swelling of guard cells over the course of a normal day, what you see is that early in the morning, potassium is the main contributor. But later in the day, the main thing that's accounting for the osmotic conditions that lead to the swelling of guard cells is not potassium, but it's sucrose. So why does it make sense in the context that we were just talking about, the chloroplasts and the guard cells? That's where most of that sucrose is coming from. Why does it make sense that sucrose doesn't contribute early in the day? Amy?
There's not that much sucrose when you just started photosynthesis.
Okay, so sucrose takes a while to accumulate. But the accumulation, the amount of triose phosphate that's made from photosynthesis, is by the time you get over here, there's enough triose phosphate to make sucrose. So that could contribute to the total amount, but the cells are completely capable of making sucrose early in the day. What else? What else do chloroplasts need to do with the triose phosphate that they make in photosynthesis? What else happens to it besides sucrose synthesis? Yeah, that's exactly it. You got to store some starch away. Remember, we said that there was an obligate pathway for starch storage in all chloroplasts to make enough starch so that the cell can have carbon available for use at night. And that is presumably why sucrose accumulation does not occur early in the day. What's happening in those chloroplasts early in the day is virtually all of the photosynthetic product is going to starch. It's not until that starch pool gets filled up sufficiently that the products of photosynthesis are used to accumulate sucrose. Okay. If you inhibit sucrose synthesis, that does not prevent the stomates from opening, because the potassium-dependent opening will continue through the day if you prevent sucrose from being synthesized. Okay, so the sucrose is replacing potassium, presumably because under normal circumstances, once you filled up the starch pool in the guard cell chloroplast, sucrose is going to be made anyway. Right? If sucrose isn't made, then potassium will continue to keep the stomates open. So an interesting question to ask would be, what is it that the cell senses that it can allow the potassium accumulation to go down as the sucrose concentration goes up? How does the guard cell sense this balance in order to keep the stomates open throughout the day? Another interesting thing to think about for an exam question. Okay, so let me reiterate then about guard cell photosynthesis. The chloroplasts that are in the guard cells are completely capable of photosynthesis, but they're not capable of the same level of photosynthesis that mesophyll cells are. Guard cells are always net importers of carbon. Right? So when you think about what's going on, when you're asked to use photosynthesis in the guard cells to interpret what's going on there, keep that in the context that the carbon partitioning within the guard cells is completely different than what it is in a mesophyll cell. Okay, questions on this before we go on? You're all looking a little sleepy again. Got a minute. We're going to give you a break. Who knows what plexers are? Plexers are word pictures that have a meaning based on the positions of the word. So how about double helix, right? Okay, how about this one? Gibraltar Cornell. This is one of my favorite ones. Caught between a rock and a hard place. Okay. Good, I got your back. Let's go on to talk about blue light photoreceptors.
Are there any ones related to plants?
I've got a ton of them, but they're not in my head. They're on paper in my desk up in my office. When I used to teach introductory biology, I did one of those every day in the middle of lecture to sort of, bring the students back from wherever they were. Okay, so we want to think about the receptors that account for these blue light phenomena. And as I pointed out, one of the ways to do this is to look for mutants that are incapable of blue light responses. So we saw back here at the beginning, in Arabidopsis, the high 4 mutants, which show no blue light bending of the hypocotyl in the dark-grown seedlings. Okay, so this tells us that there's something wrong with the blue light signal transduction pathway. Okay, so the high 4. First of all, the high mutants were a class of mutants that were discovered because they don't show light-dependent phenomenon. We actually talked about the high 1 mutant when we were talking about Phytochrome. It's missing some Phytochrome stem elongation phenomenon. High 4 turns out to be related to a blue light response. Okay, so in the high 4 mutants, there's no bending or decrease in stem elongation. So in Arabidopsis, when they sequenced the gene that accounts for this high 4 mutation, they found something interesting. They found that forgot to bring some big chalk. They found that the gene that encodes the high 4 is very similar to DNA photolyase. Who's taken biochemistry or genetics and remembers what DNA photolyase does? Nope. Okay, so DNA photolyase is a DNA repair enzyme that repairs pyrimidine dimers. So UV light can cause the formation of pyrimidine dimers in the DNA. And DNA photolyase is an enzyme, very interesting enzyme, that repairs those in a light-dependent way. Blue light is required for this enzyme to go through its catalytic cycle. So that's interesting, because if we have a mutation that gives a response to blue light in terms of bending of the cholia, or bending of the hypocotyl and changing the rate of hypocotyl elongation, that DNA photolyase is also a blue light requiring enzyme, suggests that this may have the same pigments. And the pigments that are present in DNA photolyase, there's two of them. There are flavins. There's FAD that's present in DNA photolyase. There's also a pigment called pterins. We've heard about pterins. I think I got pictures of them here. Oops. Yeah. So here's a pterin, and here's FAD. They both absorb blue light. They actually both have one of those sort of three-fingered absorption spectra. Pterin we heard about because it's also a component of nitrogenase enzyme. It plays a role in electron transfer that goes on in the reduction of nitrogen gas to ammonia in the nitrogen fixation process. Okay, so here are two potential candidates for the light-absorbing pigments in the high 4 mutant. Well, once they got this far, they wanted to distinguish high 4 from high 1, since high 1 was a Phytochrome mutant. So they changed the name of this photoreceptor. They called them cryptochromes. And high 4 became Cry1. Same mutation. They just changed the name because they wanted to distinguish the high 4 mutant that affects a blue light receptor from the high 1 mutant that affects a red light receptor. So now it's called Cry1. Encodes the same protein. It's the same gene. So it's got the similarity to the DNA photolyase. So that gives some information about what the receptor is, but that's pretty much where our information ends. In DNA photolyase, blue light causes reduction of the flavin. So the flavin picks up an extra electron in DNA photolyase. There's no evidence that happens in the Cry1 protein. The role that the pterin plays in it, we don't know. The one thing that we do know about these guys is that, like Phytochrome, blue light causes autophosphorylation. And it also causes changes in cellular localization. But again, that's pretty much where our understanding of what's going on with Cry1 mutants stops. There's a few more details in the signal transduction pathways, but how these contribute to changes in membrane potential that lead to changes in stem elongation are not really known. Yes?
And the Cry1 mutant that has DNA phosphorylase for...
No, DNA photolyase. It's not, the Cry1 mutant is not DNA photolyase. It is similar to DNA.
Do they still have the photolase?
Oh, yes.
That one then
Yeah. Plants or organisms that live in the light, if you mutate DNA photolyase, they die. Yeah. That's a lethal mutation. Okay, interestingly, one other thing that's important is that the classes of things that we've talked about that blue light receptors deal with in plants, stem elongation and bending of the hypocotyl. We've talked about stomates. The other thing that they do, we've talked about, but we didn't relate it to blue light, and that's chloroplast movements. Chloroplast movements in response to light, so that they distribute evenly across the top of the cell in low light and accumulate along the edges of the cells in high light. In the high 4 mutant, they don't do the stem elongation, but they have normal stomatal function, and they also have normal chloroplast movement. Okay? So that tells us right away that there has to be other blue light receptors that are present in plants. Right away, they identified a second blue light receptor, Cry2, which also plays a somewhat weaker role in stem elongation, but has a more important role in cotyledon expansion. So cotyledons expand pretty quickly once the plants see the light. And so Cry2 seems to play a more important role in that. But Cry2 still has normal stomatal function and normal chloroplast movements. Okay, so there's got to be some other class of blue light receptors that are important. And let's erase this down here. The other mutations that have been discovered are the Phototropin mutations. The phototropins have no chloroplast movement, or let's say altered chloroplast movement. But they have normal decrease in stem elongation and normal stomatal function. So the genes that are encoded by the phototropins, there's two genes that have been mutated. They're Phot1 and Phot2. Phot1 and Phot2 have different effects on chloroplast movements in response to light. One of them primarily affects the response to low light, where the chloroplasts spread out evenly, and the other affects the response to high light, where the chloroplasts accumulate along the edge of the cells. But there's no effects on stem elongation or movements in the movement of the guard cell movement, guard cell expansion in response to blue light. Do these guys, the Phot mutants? Yeah, so if you expose Phot mutants to excess light, they don't do as well. If you give them really high light, they die. Sort of, Ithaca light, they do just fine. So remember that what do those chloroplast movements do for the plant? Yeah, they basically protect the plant against the consequences of excess light absorption by the chloroplasts shading each other, right? So you would expect then that effects of those mutations would be most pronounced under high light conditions, right? And under low light conditions, they may show no phenotype at all. And that's pretty much the case, okay? Okay, so now we've got two classes of blue light receptors, but we still haven't accounted for stomatal function, right? We don't know a lot about, we know less about the proteins and the absorbers of the phototropins than we do about the Cry1 and Cry2 mutants, right? We know that you can induce mutations in specific genes, but we don't know very much about the proteins and what they're interacting with yet. Okay, so we need to finish up by thinking about what's going on in stomates. And let's see. This is what I wanted to go to. So this is a picture from your text that shows the red line is light intensity over the course of a day. The green line is the amount of zeaxanthin that accumulates in mesophyll cells. And remember that zeaxanthin it's a carotenoid that we talked about when we talked about photoprotection. That zeaxanthin tends to accumulate in plants that are exposed to excess light. And the zeaxanthin plays a role in dissipating absorbed light energy before reactive oxygen species can form. Okay? So not particularly surprising that as light intensity increases, that the amount of zeaxanthin that's present in the mesophyll cell also increases. And as the light decreases at the end of the day, it goes in the opposite direction. If you look at zeaxanthin in the guard cells, the amount of zeaxanthin that's made per chlorophyll is much higher in the guard cells than it is in the mesophyll cells. And this was the first clue that Zeiger, the guy who writes the textbook, took to suggest that zeaxanthin may be playing a role in regulating stomatal aperture in guard cells. A lot of you asked questions about this because what was in the textbook didn't make a whole lot of sense. And I completely agree with that. I think it's fair to say that Zeiger has had difficulty pushing this zeaxanthin control of stomatal aperture in the real, the scientific literature. But in his textbook, he can write whatever he wants. And so you notice this was one of the longest sections in this part of the this lecture topic. It's because this is, I guess, close to his heart. But I think it's fair to say that a lot of the stuff that's in there is speculative at best. And let's start with this figure, because one of the things that we talked about is that guard cells can't produce a lot of photosynthate, but more importantly, guard cells are never exporters. They're always net importers. So there's no normal sink for the products of photosynthesis in guard cells. So if you were comparing excess light absorption in a mesophyll cell versus a guard cell, which one, for the same light intensity, would you expect to be more susceptible or more prone to excess light absorption? The mesophyll cell or the guard cell? Guard cell, why? Its chloroplasts are different. So? Okay. So remember that the key thing I was putting emphasis on is that guard cells are never net exporters. They have no mechanism of exporting sucrose. So the guard cell's making sucrose over the course of the day. What's that sucrose going to do to the amount of photosynthesis that happens in the guard cell? It's going to slow it down. And if it slows down photosynthesis at the same light intensity, what's going to happen to the amount of excess light absorption? So what would you expect for the amount of zeaxanthin in the guard cells to happen? It's going to go up, right? So a number of people have shown evidence that the higher zeaxanthin content in guard cells is a consequence of the fact that the guard cells can't do much with the photosynthetic products they produce. Nevertheless, that doesn't seem to have impaired Zeiger from going on and trying to produce data that talks about the role that zeaxanthin plays in guard cell photosynthesis and stomatal opening. Now, one of the interesting things that he's shown is that there's a mutant called NPQ1. NPQ1 is a mutant in the enzyme that produces zeaxanthin. So it's not like somewhere way back down in the carotenoid biosynthesis pathway. It is the last enzyme in the pathway that synthesizes zeaxanthin. So NPQ1 cannot make zeaxanthin. So if we compare stomatal aperture associated with variations in blue light in wild type, in the NPQ mutant, and in the PHOT1 and PHOT2 mutants that affect the phototropisms, it's pretty clear that the NPQ mutation is inhibiting stomatal opening. So that certainly supports his hypothesis that the inability to produce zeaxanthin plays a role in the ability of the stomates to respond. No question about that. That's pretty clear. But this is the model that Zeiger presents at the end of the chapter to talk about the role that zeaxanthin plays. And a couple things that are important. First of all, because zeaxanthin plays such an important role in photosynthesis, it's been studied pretty greatly. And zeaxanthin is located in proteins that are in the thylakoid membrane. All the zeaxanthin in the cell is located in proteins in the thylakoid membrane, even in guard cells. So what that suggests is if zeaxanthin is the light sensor, then the place where that light sensor is functioning is in the thylakoid membrane. Right? So what that means is if the end result of that signal transduction pathway is changing the activity of the proton pump in the plasma membrane, then there has to be some mechanism of getting that signal out of the chloroplast into the cytoplasm and then bring about the effects that we've described as far as the proton pump is concerned. And so a couple of you asked the question about, so what's all the COP and PHOT and CRY mutants got to do with this? Well, we don't know. There's no idea whatsoever. What has all this stuff to do with Calvin cycle and regeneration of ATP got to do with it? Well, it's pretty much speculation. So I don't want to discourage you from reading that section of the chapter, but I think it's fair to say that knowledgeable people in photosynthesis don't give a lot of credence to this hypothesis, at least not yet, because there's not enough data that really supports it, and there's a lot of questions that are still there. That doesn't mean it's wrong. It just means that compared to some of the other things that we've been talking about, it's more speculative. Okay, we're done. Yes, of course you can ask questions.
Like, what evidence is there that says that his theory might not be correct?
Oh, okay. So what are some of the other things that he talked about in the textbook that he had a long list of bullet points that supported the hypothesis? What was the last one that he spent a whole section of the chapter talking about? The orange carotenoid protein that's in some photosynthetic prokaryotes. So that protein has been sequenced. If there was a similar protein in Arabidopsis, do you think that would have been identified by now? Yeah, because we have the whole sequence of the Arabidopsis genome. There's nothing even close. Right, so I mean that's an important one. The fact that the green light photoreversibility of stomatal opening is a really interesting phenomenon. So you put blue light on and the stomate's open. You put green lights on and the stomate's closed. Does that mean that a single photoreversible pigment is controlling that? Not necessarily. It could be a blue light receptor that opens it and a green light receptor that closes it. Just like PHOT1 and PHOT2, one of them takes care of chloroplasts spreading out evenly in dim light, and the other one takes care of chloroplasts aggregating on the sides in bright light. It could be two completely different proteins. Right? So that's one example of data that he uses to support his hypothesis that really doesn't support it very well. Okay, so like I said, I don't want to leave you with the feeling this is wrong, but I do want to leave you with the feeling that you shouldn't feel bad if it doesn't make sense to you, because that's the case for lots of people. Right? Unfortunately, it's going to be the case for a lot of things that we're going to talk about because we just don't have enough details. We know that there are genes that control these phototrophic responses in terms of chloroplast distribution. But we really don't know anything about the proteins, the chromophores, the signal transduction pathway. Grace, do you have a question?
Yeah, what about, it just seems very, like it's a lot of work to put zeaxanthin in the membrane rather than just increase in the concentration and, just generally inside the lumina of the chloroplast. So I'm just wondering why, it just doesn't seem to make sense that why would the plant be putting all the work into putting something in the cell membrane?
It's not putting it in the cell membrane. The zeaxanthin is located in proteins in the thylakoid membrane. So, and in fact, I didn't mention it, but it's an important thing to keep in mind. The zeaxanthin is not being synthesized de novo from isoprenes. Remember, carotenoids are terpenes. So they're not being synthesized de novo. Basically what happens is there are two carotenoids, violazanthin and zeaxanthin, that are interconverted by this enzyme that's encoded by NPQ1. Right? So in the dark or under low light conditions, all the zeaxanthin exists as violazanthin. Right? So in the light, you convert the violazanthin to zeaxanthin. In the dark, you turn it back the other way. So it's not making zeaxanthin de novo. It's not making the proteins that bind it. It's changing it within that protein. And as we talked about when we talked about photosynthesis, that change from violazanthin to zeaxanthin turns on new mechanisms that basically take light energy in the antenna and dissipate it as heat to prevent that light energy, the excess light energy, from causing reactive oxygen species and things like that. Okay, so this part is well understood in terms of the role it plays in photoprotection. The suggestion that zeaxanthin also plays a role as the light receptor for stomatal opening and closing is the new part of this. And it may very well be correct. It's just that the information, the evidence that's available now is questionable. We'll just have to wait and see. 10 years ago, we didn't understand any of this stuff either. That's what most of the graduate students who worked on in my lab figured out how that worked.